All right, so this is the third and final part of this video lesson for this week of homeostasis. The first part was explaining to you how the next 5 weeks are going to kind of look. The second part was all about thermoregulation and introducing to you what homeostasis is about. Hopefully you work through the worksheet and the PowerPoint to figure out what we deem important. And this last part is on osmoregulation. And we're going to focus on human osmoregulation. But we need to start from the beginning. So, I don't know how many of you read stories, but one of the stories that talks about pirates and anybody who's trapped on sea, there's this ancient mariner's rhyme, water, water everywhere, not a drop to drink. And what this mariner's rhyme is telling us is that this person at sea has all this water everywhere. They look around, water, but if they drink it, it will actually hasten their dehydration and their ultimate doom. Why is that? Right? Why is it that we cannot drink salt water? There's a story of these youths. Imagine you trying to have some fun. So pre-COVID, you go out on a Friday night and you go over to a friend's house, maybe see a movie, walk around a park or something. But imagine you lived in the Polynesian Islands. So Polynesian Islands are here somewhere between, Australia and the Americas, right? There's Hawaii over here, and you keep on going west, and you'll get to the Polynesian Islands. So you're a teen, and you just want to have a good time. So a party's happening, and then you decide to take the canoe back home, right? And that's what they do. So they hop in the canoe, and they row to the next island. But somewhere along the line, something happened, and they went off course. And look at this ocean. Even though there's an island right there, and there's an island right there, there's a lot of water here. And so going off course is not, I guess, unheard of, especially if a storm or something like that were to come up. So there was this group of teens, and I think it was 3 boys, and they got lost. They were lost at sea, and they were given up. They assumed that we couldn't find them anymore. And what had happened was they were stranded at sea for 50 days. So what do you do to survive at sea for 50 days? What do you eat? What do you drink? They were kids of the island. They knew how to take care of themselves. They were able to capture birds and use the liquid in the bird meat to keep themselves alive. They were able to do certain things that after 50 days, they were rescued by a passing transport ship. It was just luck. The ship wasn't looking for them. They just happened to see these boys stranded at sea, and they picked them up. So I would encourage you to take a look at some of the links I put in the Google Doc to look at some more of their story, because it really is quite remarkable. And there are quite a few stories of people being trapped at sea for a long period of time and then being rescued. And then hearing their story of how they did survive, what they drank, and all sorts of things is really quite remarkable. It really puts into perspective some of the things that you might undergo. So, can we survive by drinking seawater? At the end of this lesson, you should be able to tell me emphatically no. You can already say no, but do you understand why? And that's what we're exploring in this lesson. So there are some organisms that are able to maintain their... So you should be taking the Nearpod. If you haven't done the Nearpod activity exploration, you should pause the video, go to Nearpod, and there's a bunch of questions I was going to ask you. And those questions, they are around osmoregulation. So there are 2 words, osmoconformer and osmoregulator. An osmoconformer is an organism that is able to maintain, correction, it is 428, go back. There are 2 terms, osmoconformer and osmoregulator. One represents homeostasis and the other one doesn't. So can you differentiate the 2 words? A conformer, its water balance changes, or its osmolarity changes with the environment. A regulator regulates its water balance. Now, let's take a look at this fish here. Work with me. This is how I draw a fish. And I'm going to use blue to represent the water and green to represent solutes, let's say salt. So this fish here, you'll notice I have more water, I guess, and less solutes on the outside than inside the fish. So this is a fish swimming in fresh water. It's my shark. So my question to you, going way back to biochemistry days, what is happening to this fish? Let's assume that the skin and the tissue and the cell membranes of this fish are relatively impermeable to salt. But we know that water is able to move across everything readily. So knowing that information, what is going to happen here? So we have terminology of isotonic and isoosmotic and hyperosmotic and hypo. So we're using those kind of things. So if I take a look at this fish here, I have a fish that's in fresh water. All right, and there is more water on the outside than in. There's more solutes on the inside than out. So in this case, a fish that's in fresh water is going to have, and so what will happen is the water is going to move from outside in. I have to be careful with my terminology, right? I think I mentioned the word osmolarity. I have to define that for you. It's a very confusing term. So the water is going to move in. The solids are not going to move anywhere, not really. But the water is going to move in. So as the water moves in, that means this fish needs to have some kind of adaptation to get rid of the excess water. If it doesn't, then it will blow up. So freshwater fish have special adaptations that allow it to get rid of excess water. So the excess water will leave. By the same token, if I have a fish that's in salt water, the water concentration is higher on the inside than it is on the outside, so the water is going to diffuse out. So it seems counterintuitive, but a fish that's in salt water is dehydrating. It's losing water. So it means it has to have mechanisms that allow it to take water in and to release very little water. And releases all this water, it has to have adaptations that allow it to take in water. So what real adaptations would you expect? Would you expect it to release concentrated or dilute urine? Dilute urine has lots of water, concentrated urine has very little water. So you'd expect it to have concentrated urine and other adaptations that allow it to conserve water. Whereas in a freshwater fish, which is my first example here, needs mechanisms to get rid of water. So it's going to have mechanisms that are going to allow it to get rid of water. Its urine is going to be very dilute, and it's going to have other mechanisms as well. So if I took a marine fish from the salt water and I put it into fresh water, what do you think would happen? If I took a freshwater fish and I put it in salt water, what do you think would happen to that fish? So that's interesting. As a matter of fact, there are many fish that could only live in their environment. They could only live in their salt water, or they could only live in their fresh water. If a fish can only tolerate a very narrow range, we call it stenohaline. So a goldfish, for example, can only live in fresh water, nowhere else. You put a goldfish in salt water, it will die. This fish here is a haddock, and it's a fish that also requires a very narrow range of osmolarity. But some fish have a mechanism that allows them to switch back and forth between salt and freshwater water strategies. Salmon is one, right? So they go and spawn in the freshwater and then they live in the salt water. And that terminology is euryhaline. And another one is this shark. We know that if you go in the ocean, you might run into a shark. But did you know that the bull shark is able to switch its mechanisms and go in freshwater? They swim up the Mississippi River. The bull shark is responsible for, I believe, some of the most frequent shark bites because they go in the water where people don't expect to see them. Because they're, okay, I'm going to be on my lookout in the ocean, but they don't expect to see them in a river. That's unexpected. Okay, so that's stenohaline and euryhaline. Now, in terms of strategies and releasing our wastes. In biochemistry, in the ChemSketch activity, I asked you to draw 3 molecules, ammonia, urea, and uric acid. All three of these are nitrogen waste compounds, which means that as we break down protein into amino acids, and amino acids are broken down to, do whatever we need to do with them, there is a byproduct, NH2. NH2 can form ammonia. Ammonia is it's a weapon of mass destruction. You can release this as a gas to kill people. It was used in the war. It is a toxic chemical. So when it's inside your body, you must get rid of it. Fish excrete ammonia, and it works for them. In order to excrete ammonia, you need to release lots of water. You need lots of water to get rid of ammonia. So if you're a fish living in the water, that's typically pretty easy, especially if you're a freshwater fish. If you're on land, the amount of water that's needed to get rid of ammonia is really high, and it's not going to work for a land animal who's trying to conserve water. On land, you're doing everything you can to conserve water because you don't have ready access to it. So land animals use urea. Not all land animals, there's a variation, but mammals, for example, use urea. You use urea. So urea is less toxic than ammonia. It's still toxic, but not as toxic. And you do require water to remove it, but not as much water as ammonia. So it's a great strategy for land animals like mammals. But urea is still somewhat toxic, and it does require some water. Now, there's another option here called uric acid, and it is incredibly, look at this molecule here. It is really complicated. So as you can imagine, that's going to require a lot of energy to create. So why would an animal use all this energy to produce uric acid? So let me give you some hints here. Here's an animal that uses uric acid. Ostrich. Here's another one. Crocodile. Here's another one. Hummingbird. Why'd I choose hummingbird? It's too long to write. What do these animals have in common? What they have in common is that they all lay eggs. Your egg layers need a special chemical, uric acid, that can be collected in the egg for a long period of time using minimal water. Urea does not allow for this. Uric acid does. So your egg layers have another adaptational nitrogen waste. And that uses minimal water but requires lots of energy. So the trade-off works for those particular animals. Now, imagine you were in a contest, and that contest said that if you can drink some water, as a matter of fact, if you could drink the most water without going to the bathroom, you could win a PlayStation 4. Right? You could win a VR set. For a lot of people, it sounds like a really easy contest to do. So there was this contest, and in this contest, these radio hosts, what they did, I want you to listen to this link. So the link's in the Google Doc and the worksheet, and it's under I will role play a radio host. The radio host, what they did is they got a bunch of people and they said, okay, if you can drink a whole bunch of bottles of water and not use the bathroom, and whoever is able to hold out the longest will give you a Wii. And it turns out that this one lady who didn't even win, she ended up undergoing this thing called hyponatremia. And this is unfortunately, this is where you overdose on water. So you could overdose on anything. So watch or listen to the YouTube video, look what happened, do some exploration around this. But if you didn't know this, if you did not know this, design, slide size, widescreen. Let me just get this here. Pretty much everything has a lethal dose. So watermelon, for example, if you were to have 130 cups of watermelon for an average male, then that would be enough to overdose and potentially kill you. It's a lethal dose. Coffee, 70 cups. I mean, the lethal dose of a lot of things are quite large. Dark chocolate, 98 cups. Nutmeg, 27 cups. So there are lethal doses for regular foods Typically the lethal dose for things like uranium or polonium or radioactive materials is very low, and for certain chemicals like magnesium and lead is also quite low. But even water has a lethal dose, and that's the take-home message from this. All right, that brings us into the kidney proper. So this is the place where the majority of the lesson is focusing. As you go through the PowerPoint, you're going to notice a lot of terminology around the kidney. So you should be familiar with the kidney, the anatomy of a kidney. The kidney is our osmoregulatory organ. It's what allows us to balance our water. The kidney, like many organs, has various regions, and two of the regions that we use to describe the kidney include the cortex and the medulla. Whenever we say the cortex, that typically refers to the outside of the organ, and the medulla typically refers to the inside. So we'll see this with the brain. The cerebral cortex is on the outside, and the adrenal medulla is on the inside. So these are common names that are used in descriptions of organs. And so the kidney is filtering our wastes, or typically our ammonia waste, out of our blood. That's its job. In order to do this, it needs a system, an effective system. And the unit of the kidney that allows this to work is called the nephron. The nephron is made up of very specific parts. I'm going to draw the nephron, even though you have the graphic, just so you can see me kind of going through the process. I'll make the video a little bit longer, but it's probably worthwhile. So as I try to draw this, let's use black, I guess. So the nephron itself, it's a tube, right? And this tube is going to collect stuff from the blood. And unfortunately, I'm using this piece of software that has a smart notebook logo here on it, so it's going to get in the way. So what I'm drawing here are tubes. Right. And then we have this blood vessel that comes in. And if you take a tube and you put it into a knot, there'll be a lot of pressure built up in this knot. And so as the blood comes in, this blood vessel, which we're going to call the afferent. So afferent's another common biological term that refers to things coming in. Efferent is when things are leaving. So the efferent blood vessel brings it in. This bundle here of blood vessel that's all in a knot is known as a glomerulus. And so the blood is under a lot of pressure. And the glomerulus is porous, so there's a lot of holes in the blood vessel here. So because it's under so much pressure, pretty much all the liquid and anything that's small enough, like a strainer, that's able to leave the blood will leave. And it'll all come into the nephron. So everything I've drawn in black here is a nephron. The first part of the nephron here is called the Bowman's capsule or the nephron capsule. And that's where all the liquid comes in here. So all the liquid's in here. So the filtrate. The only thing that's going to be left behind in the blood, for the most part, is anything that's too big to leave. And so that's your red blood cells. So now, all this is sitting here in this first part of the nephron, which is called proximal tubule. Proximal means proximity, close. This loop here is called the loop of Henle. And the loop of Henle is made up of the descending limb, the loop, and the ascending limb. Notice there's a thick part here, right? This is thick, and there's a thin part here. So this is a thick of the descending, thin of the descending, thin of the ascending, thick of the ascending. You'll notice that I have this is not equal to here. So the thick limb on the up starts later. And this part of the tubule here is called the distal tubule. So these are the parts of the nephron. We have the blood vessel, glomerulus, and it leaves. And then we have the nephron itself with the Bowman's capsule, the proximal tubule, the loop of Henle, the distal tubule. This blood vessel, I have to be very careful. So in most of my graphics, I'm not going to draw it properly. But this blood vessel, some of it will actually go around and it will meet up with another blood vessel that's on this side, and it'll leave and go. Okay, so this blood vessel here, even though I draw it, and I'm not going to have it in my future diagrams, this blood vessel is all over the place. And so it means that there's blood supplying the tissue outside the nephron as well as the nephron itself. I normally will not draw these blood vessels on just because it really makes the diagram difficult to understand. Okay, so those are your parts of the nephron. Now, how does the kidney work? The kidney is very interesting and is very effective. And what's going to happen is the kidney is able to clean out your blood effectively. And the analogy that we use for this is called the closet analogy. So if you're fixing your closet, you might say, okay, it's just a mess in there. I'm just going to take everything out. And once you empty everything out, then you go through and you pick out what it is that you want to keep, and you put it back in your closet. And your closet, maybe you're moving or you're going away to university or something like that, hint, hint. And then you're like, okay, I have too many things in here. Let me just go back and take these last couple of things I actually didn't need out. So the kidney works in exactly the same way, using the same analogy. So what I'm going to do is I'm going to use this last bit of the lesson here to explain how urine is formed. So I'm going to work my way through it. I'll give you some links that you can also follow, and hopefully between my explanation and those links, you'll get a better understanding of the kidney. I'm also going to go back to that program that was kind of flaking out on me a moment ago. So I'm hoping that it will work for me. There we go. Okay, so if we look at this diagram, what you'll see is the afferent, you'll see the afferent blood vessel coming in. You'll see the Bowman's capsule. You'll see the efferent leaving, and remember, it's going all around these blood vessels. I'm just not showing it because it gets way too complicated. The nephron, the Bowman's capsule starts in the cortex, in the outer part of the kidney, and then the loop of Henle dips down into the medulla. And then it comes back up again into the cortex, and the collecting duct, which I did not show you, it's the final part, it dips down in the medulla and it leaves out of the kidney. Okay, and I'll show you that at the end. Okay, so the blood's coming in. So in that blood we have amino acids, and we have proteins and vitamins and urea, which we don't want, and uric acid, believe it or not, which we don't want, a little bit of ammonia even that we don't want. There's sodium and chlorine and bicarbonate and water and all sorts of things. Maybe drugs, penicillin or something like that inside your body. All these things are in your blood. So when it gets to the Bowman's capsule, everything that's large enough to leave will come out. So I'm using this little dot here to represent everything coming out. And so out it comes. So this very first step is called filtration. Everything leaves in filtration. Then what will happen is your body, it doesn't actually want to get rid of everything. It doesn't want to get rid of the glucose. The glucose we want to keep. It doesn't want to get rid of the sodium and the chlorine and the bicarbonates and the potassiums. There's some things it wants to keep. Some of those things to stay inside our body. So anything that we want to keep inside our body will get reabsorbed. So I'll use green here to represent being reabsorbed. So we're going to reabsorb some glucose, and we're going to reabsorb amino acids if they're small enough to fit through, reabsorb some sodium and chlorine and bicarbonates and potassium. All these things are going to get reabsorbed back into the body. Right? So they get reabsorbed back into the efferent blood vessel that's going past, as a matter of fact. Okay. Now, in addition to this, we also are going to follow the filtrate as it goes to the proximal tubule. Now this is where I have to review our terminology. Osmolarity. So osmolarity refers to the ability to drive water, to make water move. The higher this number, 300, the higher the osmolarity. But it doesn't mean that the water concentration is more. What it means is that it has a greater ability to make the water move. So actually, a higher osmolarity typically means that the solute concentration is higher, because then, therefore, there's a greater potential to move the water. So this filtrate, even though there's a lot of other things inside of it, the majority of it is water. The nephron is all about conserving water. We want to conserve water, keep the water in our body, because we're on land or you're in the water and you need to not lose it, otherwise dehydrate and your systems won't work properly. Inside this filtrate, there are other things as well, including yellow I'm going to use to represent urea. And I'm going to use these dots here to represent my salts. Okay, so I'm going to be done with that. So as we go down, this descending limb, the descending limb is impermeable to salts, but it is permeable to water. So water will leave. So as the water filtrate and the salts and all that stuff moves down the descending limb, water is reabsorbed. So let me grab my arrow tool here. Arrow. We'll use blue this time. And water gets reabsorbed. So as we go down in the interstitial tissue, so this is the tissue outside the loop. Up here in the proximal tubule at the top is around 300. But as we keep on going further and further down into the medulla, the osmolarity gets higher. As we go down here in the loop, the water is reabsorbed from the loop of Henle, the ascending loop, into not the interstitial tissue, but into the blood vessel that happens to be here that you can't see. I didn't draw it because it gets confusing. So as the water filtrate moves down the loop here, more water is removed. As water is removed, this gets more concentrated. So I go from 400 here to 700 here to 800 here. So inside, the osmolarity is also getting higher, but outside, the osmolarity is always higher, which causes the water to be reabsorbed. This is what we want. And then finally, we get to the bottom of the loop. So at the bottom of the loop, we kind of have a highest osmolarity around 1200. And at the bottom of the loop, the concentration of the salts is very, very high. And so now it's going to start moving back up the loop. And as we move up the loop, the salts here are so concentrated, they're just begging to diffuse out. They couldn't diffuse out on the descending limb, but they can diffuse out on the ascending limb. So they couldn't diffuse out down here, right? You couldn't diffuse here. But now finally in this area, they can diffuse out. And diffuse out they do. So on the ascending limb is now permeable to salt. So salt is now going to start diffusing out. And some of that salt that diffuses out, some of it will stay in the interstitial tissue, causing the tissue to have a high osmolarity, which is good. That's what we want. So this tissue over here, the salt leaving here, causes the osmolarity of the tissue to be high, which causes the water over here to be reabsorbed. This system is countercurrent. As it goes down here, water is reabsorbed. As it goes up here, salt is released, which allows the water to be reabsorbed. These two rely on each other. This water reabsorption relies on the salt being released into the tissue. Right? Eventually you get to a point where there's going to be a balance between the osmolarity of the filtrate and outside. The osmolarity is going to be the same. So it's going to be 300 on the inside and 300 on the outside. And at that point, the active transport mechanisms start to kick in. And the active transport mechanisms will then begin to actively transport out the sodium. So the sodium is going to be in a higher concentration on the outside than it is on the inside, but that's okay. It's just going to start pumping it out actively. So we're spending energy to actively transport these things out of the tubule. What's going to happen is the osmolarity here is actually going to drop because there's not as many solutes. So now we have a very low osmolarity of around 100 in the distal tubule. And so as the filtrate moves along this, let me use blue here. As the filtrate moves along here, eventually we get back to the collecting ducts. In the collecting ducts, this is where the distal tubules from many nephrons dump in. And the collecting duct is going to go down into the cortex. Now, remember what happened in the cortex before. As we go down, the osmolarity of the tissue is increasing, which means that water will be reabsorbed from the collecting duct into the interstitial tissue? No, into the blood vessels, which I didn't show you. Remember that afferent blood vessel is wrapped around all the collecting ducts, all the loops. So this water is going to get reabsorbed from the collecting duct into our blood supply, and it's kept. We conserve our water. This is excellent. And at the same time, the salts, specifically the urea, is going to get concentrated. Okay? And so now it's super concentrated. Now let's just follow this again. I'm going to go back and around. So initially, the blood was filtered. We end up with this filtrate. Glucose, amino acids, and various things were reabsorbed back into the body. And that left us with things that were unwanted, like urea and some salts. They went down the loop. And as they went down the loop, which is permeable to water but impermeable to salts, the water gets reabsorbed. This causes the filtrate to become higher and highly concentrated. At the very bottom, we have such a high concentration of salts, they are ready to leave. And finally, in the ascending limb, they will be diffusing out. Some will go into the interstitial tissue, which will increase the interstitial tissue's osmolarity, allowing the flow of water to be reabsorbed. Some will be reabsorbed back in the blood supply as well. So that's reabsorption down here as well. The filtrate keeps on moving up the ascending limb into the distal tubule. There are sometimes things like antibiotics, your penicillin. I don't know if you guys have ever been on penicillin, but if you are, your pee smells different because your body actively excretes penicillin. It gets rid of it. So sometimes there's things inside your blood that didn't come out properly in the filtration or got reabsorbed, and so your body will secrete it. And so antibiotics and things like uric acid will get secreted from the blood back into the proximal or the distal tubule. When we talk about pH balance, the pH is also balanced in this area. So hydrogen ions are also secreted from the blood into the nephron to help balance out the pH of our blood. All right, so this filtrate is moving along, and now it gets in the collecting duct where it goes down to the cortex, and as it goes into the high osmolarity of the tissue down here, water is reabsorbed. The uric acid and the urea specifically is now super concentrated, and most of that also gets excreted. So it moves down the collecting duct into the ureter, and off it goes. We excrete it down to the bladder. Some of this, though, comes back into the tissue. So some of the urea, a little bit, a small amount, comes back into the tissue. What purpose could that serve? Well, urea is a salt, actually. And so as the urea comes into the tissue, it helps increase the osmolarity of the tissue, which helps this countercurrent multiplication of water being reabsorbed occur. And then some of that urea might then be secreted back into the ascending limb, and then it gets excreted the next time around. That is the process of urine formation. That is a lot of information. You will notice on the PowerPoint, I've given you 2 slides that really talk about this concept. This one here talks about it in general, and I give you this table here that explains the relative concentrations of the plasma, the filtrate, and the urine. And you'll see this graphic. I took a screenshot and put it in the PowerPoint. And then here is a very detailed version of how urine is formed. Urine, when you look at your pee, I know it's easier for guys to look at it than girls, but if you look at your pee, white, a lot of people look at transparent pee and they're like, oh, okay, I'm good, I'm drinking lots of water. But it's not always a good thing. So the various colors of your pee, your urine, can indicate your health or if there's a disorder in your body. If you ever see red in your pee, go see a doctor. That's not good. Other colors, it depends. Okay? The control of osmoregulation, so urine formation, is an endocrine process. So we'll talk about that next week. So this is the urine formation. You should be able to now take this information along with the PowerPoints, along with the links I've given you from Khan Academy and a couple of YouTube videos, and you should be able to explain how urine forms. If you go through the document, you'll see some Learn 360 resources that will help you with it as well. Okay, so your end of week task is for you to explain to me, using a Flipgrid, this process, using the proper terminology. And that's it.